Welcome to Biomedics. In this episode, we take a focused look at the regulatory journey of human tissue-derived products under the 351 pathway. Whether you're a regulatory team, research, or a clinical development group, understanding the 351 process is essential for advancing safe, effective, and compliant biologics. Today's video walks you through every critical phase, from donor selection and tissue processing to clinical trials, FDA submission, and post-market responsibilities. This is part 1 in a broader regulatory series. In future episodes, we'll explore the 361 HCT/P pathway, combination product strategies, and practical planning tools to help your team build a sound regulatory roadmap from day one. This video is intended for educational purposes only. It does not constitute legal or regulatory advice. For guidance specific to your product or development program, consult your internal regulatory team, qualified external consultants, or the FDA directly.
Welcome. In this presentation, we'll explore the complete regulatory journey of a human tissue-derived product, from donor selection all the way to FDA approval as a 351 biologic. This pathway is guided by the highest standards of safety, purity, and regulatory compliance. Let's begin with Phase 1. Donor selection and screening. We'll start at the very beginning, with the donor. Eligibility is strictly defined by FDA guidance, with criteria that screen for infectious diseases, prion-related risks, and overall health. Informed consent must comply with 21 CFR Part 1271 and document every aspect of tissue use, testing, and storage. We collect detailed medical and social histories, perform physical exams on living donors, and run FDA-approved tests for HIV, hepatitis B and C, West Nile virus, and syphilis. Every recovery event is documented with aseptic technique, environmental controls, and proper labeling. Phase 2. Tissue processing and manufacturing. Next, let's look at what happens after tissue recovery, the processing and manufacturing phase. Tissues must be transported in validated containers with full traceability. Processing involves careful dissection, cell isolation, and decellularization, if needed, to reduce immunogenicity. Cell culture environments are tightly controlled to preserve viability and phenotype. Sterilization is validated to ISO standards, and viral inactivation steps are rigorously tested. Quality control includes USP 71 sterility testing, USP 85 endotoxins, and purity metrics such as residual reagent removal and cell debris. Products are preserved using validated cryopreservation methods, then labeled according to federal requirements. Phase 3. Preclinical studies and IND submission. Now, let's move into preclinical development and IND submission. Before any human use, the biologic must be fully characterized. We assess identity, potency, and purity, referencing USP monographs when available. In vitro assay test for biological activity and safety. In vivo models examine tolerability, pharmacokinetics, and toxicology. These data are submitted to the FDA in an investigational new drug application. The IND includes a detailed clinical protocol, chemistry and manufacturing controls, and a full preclinical safety dossier. Phase 4. Clinical trials, phases 1 to 3. With FDA clearance of the IND, it's time to begin clinical trials. Phase 1 evaluates safety in a small group of volunteers or patients. Phase 2 looks at early signs of efficacy and helps determine the right dosing. Phase 3 confirms those results in large, diverse populations. Trial design must follow good clinical practice. Data integrity is essential, and all adverse events must be reported promptly and thoroughly. Statistical analysis supports every claim of benefit or risk. Phase 5. BLA submission and FDA review. Now that the clinical trials are complete, we're ready for the Biologic License Application, or BLA. This comprehensive submission includes all clinical and pre-clinical data, manufacturing controls, facility documentation, and proposed labeling. The FDA carefully reviews the BLA with teams of experts. They may conduct site inspections and consult external advisors. If the product meets FDA standards for safety, purity, and potency, it will receive approval for marketing in the United States. Phase 6. Post-market surveillance. Now, let's move on to post-market surveillance. Approval isn't the end of the journey. It's the start of a long-term commitment to patient safety. Phase 4 studies continue to monitor the product's performance over time. The MedWatch program helps gather real-world data on adverse events. For higher-risk products, the FDA may require risk evaluation and mitigation strategies to ensure benefits continue to outweigh risks. And throughout the product's life cycle, companies must maintain compliance with current good manufacturing practices and submit periodic safety updates to the agency. Transforming donated tissue into a life-saving 351 biologic is more than a process. It's a responsibility that calls for integrity, innovation, and complete life cycle accountability. To succeed, teams must align with FDA expectations, maintain pristine data, and be prepared for continuous improvement. For more information on this video's contents, visit biomedicsconsulting.com. Thank you for watching.
Thank you for joining us in this episode, where we took a focused look at the regulatory journey of human tissue-derived products under the 351 pathway. This video was intended for educational purposes only. It does not constitute legal or regulatory advice. For guidance specific to your product or development program, consult your internal regulatory team, qualified external consultants, or the FDA directly.